Thanks. Thank you, Ruben, or whoever hit the record button too, because I forgot. Um, yeah, thanks to Hannah and Jordan and other folks for covering for me. This week we have Alex Mullens from the University of Illinois. He's an almost finished PhD in Tiffany Jaman's lab. What's your status, Alex?
I already graduated. So
Congratulations. Dr. Alex Mullens, everyone. Take it away.
yeah, thanks. So I'm going to be talking about the work I've been doing for my PhD, characterizing the pathosystem between Xanthomonas vesicola and maize. All right, so before we begin, I want to get everybody thinking about how a maize leaf actually has different habitats in it for a bacterial pathogen to colonize. So the mesophyll is one habitat and the xylem is another one. And they're very distinctive. The mesophyll has living cells, it's gas-filled spaces, relatively nutrient-rich, whereas the xylem is fluid-filled, constructed of dead cells and relatively nutrient-poor. The Most of the time, bacterial pathogens will end up evolving to specialize in colonizing either the mesophyll or the xylem. And plants, of course, would use different resistance mechanisms against pathogens in each of these different tissues. A prime example of this is in rice, where two different pathivores of the bacterial pathogen Xanthomonas oryzae cause different diseases on rice, different foliar diseases. The pathivore oryzae is a vascular pathogen and colonizes the xylem, causes these sort of long lesions that track along the veins, whereas the pathivore oryzicola colonizes the mesophyll and causes these water-soaked lesions that later become tan and necrotic, but are typically vein limited. And non-vascular pathogens in this particular situation are typically referred to as bacterial uh, leaf streaks, whereas the vascular pathogens are typically called uh, blights, like bacterial blight of rice. And it's a pattern that is somewhat conserved across or used across different bacterial pathosystems. So the pathogen that I've been studying is Xanthomonas vesicola, pathivore vasculorum. It's a non-vascular pathogen of maize. It's gram-negative and motile. It's typically characterized as causing bacterial leaf streak of maize, which is, as I mentioned, a non-vascular disease that's vein limited, but it also causes gumming disease of sugarcane, which is a vascular disease. So it colonizes the xylem in sugarcane. It's known to get in through wounds and stomata, cause these long, narrow lesions that are start out water soaked or vein limited, typical of bacterial leaf streak uh, pathogens. It was first noted to cause disease in maize in 1948 in South Africa. Somewhat interestingly, there in this first report, it was described as causing a bacterial wilt disease there. And then it pretty much stayed in South Africa for a long time and then was emerged in the mid 2010s in the Americas, of North and South America. So when our group started working on it, we noticed that there were some unusual disease symptoms in some maize lines when inoculated with XBV. So here I'm showing a picture of what XBV infection looks like on sugarcane. And you can see it looks a lot like the bacterial blight pathogen of rice. It tracks along starts on the tips and edges, tracks along the veins, typical of vascular pathogen. And then here's some images of inoculated CML333 in the field, and it has very similar looking lesions to what you see in sugarcane, where they start along the tips and edges, sort of track along the veins. Some of them even look like they're wilting a bit. Additionally, some first reports from Brazil show some atypical disease symptoms as well. So you have the typical bacterial leaf streak, symptoms here on the left in A, but then they call these severe symptoms where it looks a lot more like what we see in CML333 and has these long lesions. So we hypothesized that XVV could behave as both a vascular and non-vascular pathogen in maize, even though most of the literature up to that point had suggested that it was purely a non-vascular pathogen in maize. So when I started working with it, I screened a diverse panel of maize lines using two different inoculation techniques, a infiltration inoculation, which is what you typically use with non-vascular bacterial pathogens, where you just push the inoculum into the mesophyll of the leaf, and a cut inoculation, where I cut the tip of the leaf and dip the wound in inoculum, where I'm introducing the bacteria to the open xylem vessels directly. And what we found was that the different maize lines seemed differentially resistant or susceptible, depending upon the inoculation technique that I used to screen them. So if you look here, the CML333 was extremely susceptible to cut inoculation, but moderately resistant to infiltration. You can even see Ohio 7B was extremely susceptible, and Ohio 43 were extremely susceptible to infiltration, but somewhat moderately resistant to quite resistant to cut inoculation. Additionally, the symptoms that the two inoculation techniques induced were quite distinctive. So the infiltration induced typical bacterial leaf streak-like symptoms. They were vein limited, started out water soaked, and then became tan and necrotic. And then once the lesions formed, they stayed that size. They didn't grow continuously. Whereas the cut inoculated leaves, it developed these grayish tan lesions that tracked along the veins and expanded continuously. When I stem wounded them and injected the inoculum into the stem, the CML333 plants exhibited systemic wilt symptoms, where B73 did not. So I created a GFP expressing strain of the pathogen to try to confirm our suspicion that it was behaving like a vascular pathogen. And sure enough, it was. And we can see these A and B, these were spray inoculated. They're from different time points, and we can see that the bacteria are able to enter via the hydatodes in an unwounded leaf. In these other images here, C, D, and E, C is a cross section of a stem of one of those plants exhibiting systemic wilt. So you can see the different vascular bundles have fluorescent bacteria in them. And then D and E are cross sections of leaf veins. And E, the bacteria are streaming out under a cover slip into water. So this is the vein here. And then in D, you're looking sort of down at the cross section of the vein, and these are the xylem vessels here, proto and metaxylem. Another thing that we noticed is that the different inoculation techniques, cut versus infiltration, would cut induces pretty much just vascular colonization, and infiltration induces pretty much just non-vascular colonization. And you can see that in these images. You have a vein going down the middle of this lesion, and it's not infected, but the mesophyll right next to it is, where cut inoculation, you see pretty much just the veins being infected here. So I decided to screen a mapping population descended from B73 and CML333 using both of these different inoculation techniques. And I was able to find two different large effect QTL that each controlled resistance to XVV in that particular tissue type. So vascular disease resistance, there's a major QTL on chromosome 5, and in non-vascular infiltration-induced disease, there's a large effect QTL on chromosome 4. And then to try to confirm these two large effect QTL that look interesting, we turned to a previously existing neurosogenic line population and selected lines that had introgressions that overlapped with the QTL, large effect QTL of interest here. And so I was able to confirm here, so these were cut inoculated, this NIL 1292 is the nil that had a CML333 introgression at that chromosome 5 locus, and it was significantly more different than B73 when you cut inoculated it. Similarly, this nil 1092 had an introgression on chromosome 4 and was also significantly more susceptible than B73, similar to what we see in CML333. You can see the qualitative images here. Additionally, when I dual inoculate, so if I here I'm showing cut inoculation again, but of nil 1092 and nil 1290. You can see nil 1290 still significantly more susceptible, but nil 1092 not really different from B73. Then when you infiltrate, you see the same thing. Nil 1290 not different than B73, but nil 1092 is significantly different. So it shows that the idea that the resistance to the different tissue in the different tissue types is independent of each other, inherited independently. I also screened these other nils. These have teosinte introgressions, teosinte parviglumis introgressions at the chromosome 5 QTL, and they were also significantly more susceptible than B73. So this represented an alternate allele of that susceptibility allele of that QTL that we were interested in. And then one of these was susceptible to infiltration. So I also screened one of the teosinte nils in nil 1290. So that's the vascular susceptibility nil against Clavibacter braskensis. This is a gram-positive vascular pathogen of maize, and they were also significantly more susceptible than B73, suggesting maybe this QTL has an effect on more than just XVB, but other vascular pathogens as well. So with this data in mind, we wanted to characterize this further. And so this was sort of a flow chart of the plan that we had come up with at this point to try to figure out what was going on further. So I decided to do a dual RNA-seq experiment in which we got reads from both the plant and the pathogen. At the same time as that, I did a mirror of the dual RNA-seq that I inoculated with the GFP expressing pathogen. And so I have images of disease at the same time points that I collected samples for the RNA-seq experiment. And then part of it, we wanted to look at the differentially expressed genes within QTL to try to figure out what the genes underlying these different QTLs were. But we also were interested in how the pathogen itself behaves differently when colonizing the xylem versus the mesophyll. And to do that part, we only looked at CML 333 inoculated tissues, whereas this one for looking at disease resistance, we looked at both B73 and CML 333 tissues. So first, I'm going to talk about some of the things that sort of an interesting story that we got out of the bacterial side of this. So here I'm showing how the cut inoculation progresses across time in B73 and CML 333. And early on, there's not much to see at 2 DPI. 5 DPI, you can clearly see it in the veins in both CML 333 and B73. But between 5 and 7 DPI, B73 will develop HR-like spots along the veins. That's what these are here that seem to prevent further progression of the bacteria in the xylem, where CML 333 doesn't do that. It does have some that what look like resistance reactions or HR-like spots along the veins, but they don't seem to be effective at stopping the bacteria from progressing further. There also are these water-soaked spots where the bacteria break out of the xylem in CML 333 that we see at 7 DPI. And so here I'm showing the infiltration. And early on, to the naked eye, at 2 DPI, the infected leaves look asymptomatic. And so here the gray images are light being shown through the bottom of the leaf. Uh, so what we see is that even though it looks healthy, we can see bacteria in the leaf tissue under the microscope. But at 5 dpi, the tissue becomes water soaked. And so you can see this, with the tissue becomes translucent, more light shines through in these black and white images, just natural light. And then you can see lots of fluorescence here. And then by 7 dpi, the water-soaked lesions start to dry out, become tannin necrotic. There's also some autofluorescence, or there's halos of uh, tissue around the edges of the lesions. And so here, so A and B are close-ups of these resistance-like reactions in B73. It looks like there's autofluorescence in the cells themselves here, is what I'm trying to highlight. In CML333, there's less of that autofluorescence in these HR-like reactions. And then G is one of those water-soaked spots. F is a 2 dpi infiltration image, and you can see, so here's a stomata or stomate, and here's mesophyll cells with bacterial clumps sort of sticking to the mesophyll cells here early on, compared to later in the infection where there's lots of bacteria between the mesophyll cells and the stomate. So keep that in mind. I'm going to be talking about the sort of the differentially expressed virulence and motility genes here. Of particular interest is the motility, as we see some differences in how that plays out in the different tissues. So here, the xenic samples were just grown on plates. And so these genes down here that are colder, they are the virulence genes for the most part. And then these are some other genes. The ones I've highlighted here are flagellin and then the type 4 pilus, which is involved in twitch motility. So bacteria have two different types of motility that they utilize, flagellar motility, which they use in fluids, and they sort of swim through the fluid by spinning their flagellum. And then twitch motility, where they send out these little grappling hook-like structures and sort of crawl along surfaces. So they attach to and crawl along. What we see is that during infiltration-based inoculations, they early on they're expressing more of the twitch motility gene. Here, still some at 5 dpi, but then less at 7 dpi. But then for the flagellin, we see less expression of flagellin early on, but then more so at the later time points at 5 and 7 dpi. And so it seems like because at 2 dpi they're not water soaked yet, so there's not a lot of fluid for them to swim in. So it seems at 5 dpi, when there's fluid there for them to swim in, they start expressing more flagellar based, more flagellin and probably more flagellar based motility. Whereas early on they utilize more twitch motility and then less so as the disease progresses in the uh, infiltrated tissues. And then down here, these various secretion systems are secretion systems that they use to secrete effectors, particularly this type 3 secretion system. I pulled this one out just to show it, and they are expressed in inoculated samples at the earlier time points. These effectors are used to suppress the defenses of the plant. So it makes sense that they're particularly type 3s, that there's higher expression earlier on when there's more living plant cells in contact with living bacteria compared to later on when the uh, plant cells are dying at that point. And then when we look at the cut inoculated samples, we see that it doesn't seem to be expressing either flagellin or the type 4 pilus gene much at all. And so it seems to be entirely non-modal when it's colonizing the xylem, whereas when it's in the mesophyll, it seems to use both forms of motility at different time points. And then just generally speaking, the the virulence genes are more highly upregulated in the cut samples relative to the infiltrated samples at this time point, at the time points that I looked at. So going more with the motility story, we thought that was interesting. I can actually observe the bacteria that stream out of infiltrated tissues, and we can see that they are motile, as you can see here. Here's a GFP video, the same thing. They appear quicker in GFP because the exposure time was longer. But in the cut inoculated samples, they're not motile. As you can see, there's some Brownian motion, but for the most part, they don't swim. So I followed up with a knockout experiment, and I made some non-motile mutants of XVB by knocking out flagellin. And so here's an image of a motility assay where we have watery agar, and you can see an expanding disc in the wild type strain here where they're swimming into the agar. And then these other ones don't have that, so they're non-motile. And so we hypothesized that flagellar motility plays a role in non-vascular colonization because it's expressed in the non-vascular tissues or non-vascular colonization, but not in vascular colonization. So I inoculated these non-motile strains into T-nil1030 in an experiment. So this was that nil that I mentioned briefly before that was susceptible to both vascular and non-vascular colonization. And we found that during non-vascular colonization, the non-motile strains appeared significantly less uh, aggressive compared to the uh, cut inoculated experiment with those same strains where we didn't see significant differences. So it seems like motility does play a role in lesion size and disease in XVB for the non-vascular, but not so much for the vascular disease. So next, I'm going to talk about the efforts to try to characterize the genes underlying the QTLs. So here I'm just showing the significantly differentially expressed genes that were within the chromosome 5 QTL. This is the vascular resistance QTL. And of these, one of them in particular stood out as being, based on how it's annotated, a probable LRR. So the negative fold change here means that it was more highly expressed in the CML333 samples compared to B73. And then for the chromosome 4 QTL, I highlighted there were multiple potential genes here that have uh, potential disease resistance conferring properties here that were differentially expressed. So I decided to follow up more on the chromosome 5 QTL since vascular disease resistance and colonization was a bit more novel in this pathosystem. So here I'm showing the GO-term enrichment of the weighted gene co-expression module that contained the probable LRR. So I looked at pulled the genes that were co-expressed with that probable LRR on the chromosome 5, then did GO-term enrichment on that group of co-expressed genes. And we see a lot of interesting things here that like detection of bacterium, plant hypersensitive responses. So more things that make us think that it seems to be co-expressed with classes of genes associated with bacterial disease resistance. Additionally, when we look on the genome browser, both B73 and CML333 produce transcript variants, but they're different between the two. In particular, CML333 uses a different start codon compared to the B73 transcripts, and it has an 11 base pair deletion up here, upstream of the start codon. But if it were present in the B73 ones, it would create a premature start codon and make the gene non-functional. So I followed up with uniform mu lines, and Here it has a transposon in the third exon of the gene. And this is a little model of the B73 transcript. The plants that were, here I'm showing segregating subfamilies, plants that had the transposon insertion, which I'm indicating with this minus minus, were significantly more susceptible than their wild type siblings here, and you can qualitatively see that as well, which on the right. And then I also saw the similar thing in a bond mu population where we have a transposon insertion again in that third exon, where the minus minus individuals that had the transposon insertion were more susceptible than their wild type siblings. I've also been working to fine map that chromosome 5 QTL using populations that are descended from the nil 1290 and the teosinte nil that I mentioned previously. And the highest marker here in each of these is within the candidate gene. Within the teosinte population, I was able to narrow it down to a 10 gene interval. And then in the nil 1290 population that has the CML333 allele, I was able to get it down to a single gene interval and was able to find 4 recombinants that were actually within the gene itself. And you can see that the individual recombinant lines that had uh, that 11 base pair mutation uh, were more susceptible compared to the one that didn't here. And so we can reasonably say that this is the causative mutation here for the susceptibility phenotype we see in CML333. So as I mentioned, there's the 11 base pair deletion, the CML333 allele. The teosinte allele has an 8 base pair insertion in the first exon. So there's a plausible reason why it's the same gene in that population as well. So looking more into what this gene is, it's a lectin-like receptor kinase. It produces in its extracellular ectodomain has tandem lectin-like domains, and in the related genes that have been characterized, it forms this forms a cleft in the middle between these two lectin-like binding domains, and it implants they bind signaling proteins in this cleft. And these lectin-like receptor kinases have been characterized in other plants to have roles in various parts of immune responses, including PTI and ETI sort of responses. And here I used AlphaFold 3 to make to simulate the ectodomains of the B73 allele and what the transcript produced by CML333. And you can see that CML333's allele lacks one of these molecular-like binding domains, so probably either can't bind the same ligand as the B73 allele or does something else in the plant. I also screened a couple of these uniform mu populations with Stewart's wilt and found significant differences there as well. So maybe this candidate gene also protects or has a role in conferring resistance to other xylem-based pathogens and might indicate that it doesn't recognize something specific to XVV, but something more general to xylem colonizing pathogens. So some ongoing projects relating to this this whole project here is we're trying to figure out what it might be interacting with using computational modeling with some collaborators. And then we're also interested in exploring the allelic diversity of this probable LRR using an allelic screen population. So as I mentioned, it's a lectin-like receptor kinase. These lectin-like receptor kinases have been characterized to bind these signaling proteins called RALFs or rapid alkalization factors. These proteins themselves have a highly conserved uh, motif that is essential for binding here. And then when I simulate that interaction in AlphaFold 3, it does predict a fairly strong interaction. Whether that's meaningful or not, I'm not sure. But we're working with Hyson Kim and Carson Andor to try to put together a computational pipeline or utilize their pipeline to try to predict what it might be interacting with, whether it's an effector secreted from Xanthomonas or a signaling molecule from maize itself. And then I'm also been working to try to understand the allelic diversity of the probable LRR. So uh, in collaboration with Dr. Alvarez, we got some 53 lines from the Bezier population that have geolocated diverse teosinte accessions at that chromosome 5 QTL. And then I screened them in the field last summer. And so here I'm just showing a climatic map of the diverse sort of climates, agroclimatic regions of Mexico here. And then here are where the teosinte, the ancestral accessions of those nils that I screened come from in Mexico here. And then from those lines, about 13 of them looked susceptible qualitatively, similar to what we see with the nils I screened previously. And so this is where the blue dots represent susceptible individuals, and the red ones were resistant. So there was about, it's easier to see here. So the red ones here are the Balsas teosyntes here. They form this little cluster. It is the more northern individuals of that group that were susceptible, and then some other Mexicanas up here. I'll show that here. So we saw some susceptible individuals from every subpopulation. So we had some lines that had Diploparentis introgressions, Ligurians introgressions, Balsas teosyntes, uh, and then the three different Mexicana races here. So we had a few individuals from each one that were susceptible, but in particular interest, these Durango subpopulation or family, I think they're races of the Mexicana group were all quite susceptible. And then these are known to be especially adapted to drought environments and come from a very dry region of Mexico. And this is sort of interesting to us because CML333 was bred from material that seems to have relatively recent post-domestication admixture from Teosinte mexicana, and it actually has some Mexicana-like drought tolerance and avoidance genes that have been characterized previously. So perhaps it seems like the lines from the drier climates might seem to have the more susceptibility to XBB, or whether this is just lower disease pressure, so they didn't need it, or that it has some advantage, some role in adaptation to drier climates, is something we're interested in exploring further. So some key takeaways: maize exhibits tissue-specific resistance to XBB, and a probable LRR receptor-like kinase confers vascular resistance to XBB. And this has implications for other pathosystems as well. So I mentioned previously that XBB can colonize sugarcane, but the same species, different pathovar, can colonize and causes disease in bananas, susceptible or a bacterial wilt disease in bananas. And then another pathovar of XBB or XV causes disease in sorghum as well. So understanding how XVV colonizes maize and the genes underlying resistance could help with some of these other pathosystems as well. And then also just knowing that it exhibits sort of a different disease, sort of like this, a leaf blight-like disease, similar to what we see in rice, could be important for breeding for resistance and identifying it in the field. So I'd like to acknowledge Dr. Tiffany Jaman, my advisor, and then all the people here who have helped me along the way with this project.
Okay, thank you, Alex. There's already a set of questions in the chat, so let's go to the chat.
Okay.
So Jeff has a question that he says, but then he had another question. So go ahead, Jeff.
Yeah, I was just wondering, you were arguing about similarities between CML33 and Mexicana. Have you looked at the two Mexicana inbred genomes on maize GDB to see if they share alleles with CML333?
I have not done that myself. So there is a paper out that looked at, I think it's ZMDR01, a drought avoidance gene, where they mention that it has a Mexicana-like allele there. And then there was another paper that I read where they mentioned that the chromosome 5 region around the centromere was very similar to what they see in teosintase as well. So it's from those papers that I'm drawing that conclusion, but I haven't actually done that comparison myself.
Okay, and then John Hodge, a couple questions. So John, do you want to?
Yeah. I guess like the first question that I kind of had, which whenever I see these LRR receptors sort of pop up, I always kind of am like, wondering whether they're part of like, the multicellular program of like, how plants are putting themselves together, or if they're still acting like disease resistance factors. And I guess especially this sort of vascular or non-vascular variation that you were sort of seeing. I was kind of wondering how many of these might actually be sort of mechanical of the leaf is sort of building itself slightly differently and having like maybe a more complex internal anatomy that is providing mechanical resistance that's actually sort of making it harder for the infection to kind of take place. Like, have you been looking at like morphological characters at all in your population to try to like deconvolve that?
No, I haven't noticed any morphological differences in the nils or the mutants in terms of how they look under the microscope or anything like that, but we haven't looked in great detail. But I think you could be right there about it having something to do with how the cells are organized. A lot of the related genes in that molectin-like receptor kinase family are involved in cell wall uh, sort of maintenance and sort of monitoring it. And so it would make sense that perhaps it has something to do with uh, detecting perhaps damage to cell walls and perhaps that way. It is like I've noticed just on maize TV that it is expressed in other tissues like pollen tubes and other things. So it's certainly possible that it has other roles, or those other transcripts might have other roles too.
And I guess like uh through my like last question, which hopefully this is a simple one. The dry land accessions that are more sensitive to infection, I don't suppose those have like a leaf rolling phenotype at all if they're dry land adapted by chance. Or uh maybe you haven't like sort of like treated them in a way to like screen for that at all yet, but we're just curious.
No, we haven't treated them like under drought stress conditions yet to see if they have any kind of leaf rolling phenotype. As far as I can tell, they looked normal in the field.
Okay, and Sanshin had a question.
Yeah, yes, Alex, nice work. You have uniform mu and Bohr mu on the receptor. have you checked, you talk about maybe this chromosome 5 locus confer the resistance to Gauss's wilt. Have you checked those mutants on Gauss's wilt yet?
Uh, Not the mutants yet. So yeah, but that's something we want to do in the future. So the difference that I noticed with the nils or Gauss's wilt was relatively small. So I mean it was detectable, but it was it wasn't like as strong and qualitative as it is in XVV. So it would require probably a lot of reps in the field to do. That is something that we would like to do and test whether these mutants confer resistance to other vascular pathogens further. Yeah,
Thank you. Nice work.
thanks.
I don't see anything else in the chat. I had a question as well, Alex, which is you showed that when you look at bacterial mutants that are non-motile, that they are not compromised in vascular infection. Do I have that correct?
Yes, that is correct.
So they're able to just, once they get in there, they're able to just like float around or whatever. I'm just wondering, it's maybe really stupid, but like if they're infecting from the tips of the leaves, are they going kind of against the current in the xylem? Like is the water is flowing toward the tip of the leaf. So how, I'm just curious how if you have an idea how they're moving maybe against that pressure.
That is something that has made me wonder a lot in the past too. So it's something that we see in Gauss's wilt too. So during my master's, I worked with Gauss's wilt and made GFP strains there. And we see both basipetal and acropetal movement in the xylem for that one, and it's non-modal entirely too. So bacterial pathogens do seem to have some mechanism of apparently moving against the flow of the xylem. I'm not sure of the mechanism itself, though, in this case. From what
Must
I
be
yeah,
Or maybe just the pressure it's just not that strong and they can even sort of randomly Brownian motion float around a little bit. I don't know.
perhaps. I'm pretty sure they have the ability to attach to the walls of the xylem. Um, During guttation, so under high humidity conditions, or if you've ever been out in the cornfield early in the morning, you see a lot of little droplets along the edges of the leaves. Uh, that's guttation droplets, and the bacteria can be drawn in through those. So there's potentially times uh, when the xylem is pulling fluid in, and maybe it's not always constant flow. Is one thought of mine.
Okay, thanks, Alex. Nice job. Appreciate it. See any other questions so we can wrap up here. Next week we have Nikki Shretha from James Schnabel's lab at University of Nebraska. So thanks everybody. Thanks, Alex. See you next week.